Dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. Welcome back to AmreenaMehrajTutorials. Today we will start for class 12th and we will be taking this unit called genetics. This unit falls under both your botany syllabus as well as zoology syllabus. But at our channel, you will find all the videos pertaining to genetics under one playlist. Okay, I label that playlist as genetics. So you will find Mendel and his contribution, molecular basis of inheritance, and you will find the basic zoology portion, which includes genetic disorders and all. You'll find all these topics discussed under one heading, that's called genetics. Today we'll begin with mutations. We'll try and understand what mutations are, because that will be the foundation for understanding genetic disorders, which we'll take up first. Don't forget to like, share and comment on my channel, and simultaneously subscribe to my channel so that I can reach to more and more learners. Let's get started. My dear students, what is a mutation? What is a mutation? What do you call mutation? Any sort of change which happens to the genetic material is basically mutation. Okay, so I'll write down over here that any sudden change, because these changes are abrupt, they are sudden. Any sudden change which happens to what? Which happens to the genetic material. That change is called mutation. Now, what is genetic material? It's the DNA. And the same DNA, you will find this in different forms in your cell. So any sort of change which happens to this DNA, that is your genetic material, that change is called mutation. Okay? Now, you know that this DNA, which we are talking about right now, which we are concerned about right now, this DNA or genetic material can undergo mutation in two versions. Either you will find its amount getting mutated, or you will find its structure getting changed. So if both of the things happen, we say that mutation has happened. Let me give you an idea. When we talk about DNA, what image you get in your mind is this double-stranded DNA. This is the image which comes into your mind. And you know that double-stranded DNA has nitrogenous bases over here, which are paired with each other. Okay? If any sort of change will happen to these nitrogenous bases, if any change will happen to these nitrogenous bases, maybe some base is getting replaced with another base. Okay, maybe some particular base is getting deleted or some particular base is getting added. So the normal sequence is getting changed. That change you will call mutation. So that is the change which is happening at which level? Which is happening at the gene level. Okay, the same DNA in a cell, you will not find it in this form. You will find it in a coil form, in a compact form. I'll be giving you details about that DNA supercoiling ahead when we will take up molecular basis of inheritance. But let us get some idea over here. So double-stranded DNA is basically wrapped around histone proteins, and you will get a model which is called beads on a string model. Okay, then that is further coiled into a solenoid, which is further coiled into a super solenoids. Then you get a structure which is somewhat like this. This compact form of DNA is your chromatin, to be very simple. This is chromatin. And you will find this form of genetic material. This form of genetic material you will find in a non-dividing cell. Okay? When a cell is not dividing, you will find this form of genetic material. But when the same cell has to undergo cell division, at that stage you will find this chromatin condensed in the form of a chromosome, which you know. What is this form called? It's called chromosome. So chromosome is basically what? Technically it's your genetic material, but basically it's a compact form of your DNA. Now, where could be the mutation happening? Mutation can happen at this level, which I told you right now. Mutation could happen at this level, or same thing can happen to your chromosome. Maybe the number of chromosome can increase, or maybe the number of chromosome can decrease. If this happens, this is also mutation. Maybe a part of the chromosome gets deleted, or a part of the chromosome gets added. All these are what? They are structural changes. All these will be discussed by we people under mutation. So I'll teach you inshallah about genomic mutations, okay, where we will discuss about increase or decrease in the number of chromosomes. I'll be teaching you, inshallah, about chromosomal aberrations, where I will give you idea about chromosome being reduced in size, a part of chromosome being added. Okay, then I'll give you idea about this, where changes will happen at the gene level, which we call gene mutations. So I'll be giving you idea about chromosomal mutations as well as gene mutations. I'll be giving you detailed classification also ahead. This was just an elementary introduction I was giving you people. Okay, now my dear students, let us have an idea that who gave this concept, who introduced this concept of mutation into the world of genetics. Let's look into that, because that is also very interesting and that is also important as far as your competitive examinations are concerned. Okay, so here you must know this scientist. He is a Dutch botanist. Botanist is somebody who is studying plants. So a Dutch botanist, his name was Hugo de Vries. He is a prominent and eminent scientist. He, in the year 1901, introduced this term. So the term mutation, if you'll be asked, who gave this term mutation? This term was given by Hugo de Vries in the year 1901 when he was studying a particular plant, which is basically primrose. Specifically, it was evening primrose. Scientific name of this plant is Oenothera lamarckiana. So this is the plant Hugo de Vries was studying, and he saw that there are various variations which are inheritable, which appear in this evening primrose. Then he said that there could be some kind of change which is happening at the genomic level, which is happening at the genetic level with this plant. That's why these variations are coming up. Then he gave those kinds of changes. He introduced the term mutation for those changes. And he even used the same concept to devise a theory which is called mutation theory. Okay, considering his same work on this plant in which he was observing different types of variations are arising, and he gave a concept, maybe mutations are, changes are happening at genetic level. He used the same concept to devise this theory called mutation theory, which he used to make us understand this concept of organic evolution. So according to him, he said that evolution happens, changes happen, species could evolve from an already existing species because of this mutation. So in support of organic evolution, he presented his idea of mutation theory. Okay, then who was the scientist who gave us the scientific proof? So the scientific study of mutations was done by Thomas Hunt Morgan. He is a prominent name in genetics, T.H. Morgan. He performed a series of studies on a model organism which he took was Drosophila melanogaster. This is a fruit fly, okay, and this organism is called jackpot of genetics, because whatever is the study of genetics which is even done today, be it in terms of detecting the DNA prints and all, that all study is done considering this organism as a model. So Thomas Hunt Morgan also performed experiments upon the same organism in which he gave us the scientific proof of mutations, and he saw that mutations do happen to the organisms, and mutations are inheritable. They are transferred from one organism to another organism. He is the scientist scientist who then proposed or who then clarified to us the concept of linkage, which we will study in this chapter. He gave us this concept of criss-cross inheritance, which I'll make you understand inshallah. He also gave us this proof of crossing over, which happens during meiosis. So all these things became clear when Thomas Hunt Morgan performed this study on Drosophila melanogaster. He actually performed the scientific study of mutations. So you have to remember these two scientists. Number one is your Hugo de Varies, who performed the study on evening primrose, Ornithera lamarckiana, and you have to remember about Thomas Hunt Morgan, who performed studies on Drosophila melanogaster, which is a fruit fly, and he gave us the scientific proof for this concept of mutation. This was basic idea. Now let us straight away go to what basically mutation is. So you must have this idea that an organism, human beings, because we are concerned with humans over here, human beings have two types of cells. What are the two types of the cells human beings have? We have somatic cells and we have germ cells. So somatic word you people know, somatic cells are our normal body cells which do not have any role with the reproduction, and germ cells are the sex cells which are responsible for the production of gametes. So accordingly we can say that mutations are of two types. We can say mutations are, number one type is somatic mutations, and number second type are germinal mutations. This is one way of classification of mutations, somatic mutations or germinal mutations. Somatic mutations are those mutations which are happening to the somatic cells. Suppose are maybe our skin cells, brain cells, kidney cells, hepatocytes. They are all somatic cells, body cells. These mutations, they will not be inherited. So we'll say they are non-inheritable mutations, because they cannot be inherited by the next generation, because these cells are not participating in reproduction. But if we are talking about asexually reproducing organisms, okay, we have certain organisms which perform asexual reproduction. In those cases, if a mutation happens to the somatic cell, that becomes inheritable. But we are not concerned with these asexually reproducing organisms. We are talking about sexually reproducing organisms. In them, somatic mutations are generally what? They are non-inheritable. When we talk about germinal mutations, these mutations are happening in the sex cells. So here we are concerned with the gonads. So we are concerned with those cells which are responsible for the production of gametes. So if any mutation will happen to the gametes, that mutation is called germinal mutation, and that will be definitely what? That will be a inheritable mutation which will be transferred to the offsprings. Okay, this is one way of classification of mutations, that mutations could be somatic, mutations could be germinal. Another way of classification is that you can say mutations could be like spontaneous. Say for example, we can say mutations are spontaneous, arising as such maybe during the process of DNA replication, because the enzyme which is synthesizing DNA, that is your DNA polymerase, it has not got 100% fidelity. So there will be certain wrong bases which will be incorporated into the newly stranded DNA. So newly synthesized DNA strands may have some errors which we'll call copy error. Such mutations do happen, and they are generally not so lethal, and you can call those mutations spontaneous. They are happening as such, and they are basically the reasons. They are basically the mutations which are responsible for variations. Okay, another class could be induced mutations. So my dear students, now over here this is very tricky. Induced mutations, in which you are using some kind of agent to induce mutation in a cell. That agent could be maybe a radiation, or that agent could be a chemical which you will use to convert or to induce change in the genetic material of some cell. If you yourself try and induce mutation in a cell, that type of mutation is called induced mutation, okay? And these agents which are responsible for inducing mutations, they are called mutagens, okay? Later on, once we'll complete this topic, I will be giving you one lecture for this also. Right now, our focus is neither somatic nor germinal. Neither we are going to stress on spontaneous or induced, but these terms are there in our textbook. These terms are there which are asked in examinations also. Our major focus point is a different set of mutations, that is chromosomal mutations and gene mutations. Let us go to that now, which is very, very important. But you have these terms also discussed on your textbook, as well as they are always asked in your examinations. So now let us go to the actual important stuff of mutations. So mutations, now we will classify them into two types. Number one, I'll call gene mutations. Number second, I will call chromosomal mutations. Gene mutations we will finish first, then we will go to chromosomal mutations, okay? Let me first of all give you this classification. Under gene mutation, we will study number one, substitution. Number two, addition. Number three, I'll use this word. I have used addition word. I'll use this word deletion. Because I had used addition, either I have to write down addition or insertion. So I've used addition, so it will be substitution, addition and deletion. These three we will understand under gene mutations, and in the same topic I'll give you this concept of FRAMESHIFT mutation also, because it's related to addition and deletion, okay? This will be finishing in today's lecture. As far as chromosomal mutations are concerned, over here we have to discuss genomic mutations, which are of further of two types. Number one, you have euploidy and aneuploidy. Okay, we'll discuss both inshallah. Number second is your chromosomal aberrations, which are further of two types. Number one being intrachromosomal aberrations and interchromosomal aberrations. So under this genomic mutation, we will see change happening to the number of chromosomes. Under chromosomal aberrations, we will study change happening to the structure of chromosomes. Under this category, gene mutation, we will be studying change happening to the gene, or that is called sequence of nitrogenous bases. This is very important because you have to remember everything. Now how you will remember? Because I have earlier told you, there will be gene mutations or there will be chromosomal mutations. So if there will be change at the gene level, if there will be change with the sequence of the gene, you will study them under this category. If there will be change in the number of chromosomes, that will come under genomic mutations. If there will be structural change, that will come under intrachromosomal aberrations and interchromosomal aberrations. Even in interchromosomal aberration, we have to study about translocation and we have to study about duplication. Here we have to study about different types. So we have multiple subtypes of this category, okay? So this we will keep for next lecture. right now we will be taking up gene mutations. Let's go to gene mutation. So we will take up first of all gene mutations. Let me take the first type, that is the substitution. Okay, so my dear student, what is a gene mutation, by the way? So, any alteration, any change which happens to the sequence of nitrogenous base of the DNA, that change is called gene mutation. Okay, let me give you one idea over here. Suppose, say for example, your DNA has a sequence of ATC, GTC, CCG. Okay, this is a sequence. Okay, now you know that when we say that it's a gene, it's a particular sequence which will be coding for some polypeptide. Okay, so your DNA is having information in the form of a gene. So gene is basically what? It's an arrangement of nitrogenous bases which is having information for some polypeptide. Now, if anything wrong will happen to this sequence, there is a chance either this polypeptide will not be formed, or if it will be formed, it will be a non-functional polypeptide. So there could be different reasons. Either it will not be formed, either it will be formed, it will be so large or so short that it will be totally non-functional, or it will be carrying some different amino acids which were not supposed to be present in it. That all happens because of what? That all happens because of mutation. So gene must be clear from this point of view. Gene must be clear to you. Now let me take you to this substitution. Okay, now you will be able to understand it. Say for example, you will be having a case where replacement of nitrogenous bases occurs. So substitution is very simple. In place of adenine, you will have thymine. In place of guanine, you will have cytosine. In place of cytosine, you will have guanine. So what is happening? Replacement. A correct base is replaced by an incorrect base. So there is substitution. You do not have removal or addition. One base is getting substituted with a wrong base. That phenomenon is called substitution. Let me give you an example. Say for example, we have C in the same manner, CTG, CCT, CTG. We have a sequence like this. Okay. Let me make it long. ATG. This is a sequence which is in your DNA. So you know this basic thing that all the information which is there in the DNA, that is read, that is decoded in triplets. Okay. So these triplets are called what? They are your codons. These codons, one codon has information for one amino acid. They are very specific. Although you have certain amino acids which are represented by more than one codon, but you have to remember this basic thing that once you have this sequence ATG, it will be specific for a particular amino acid. If something wrong will happen to it, there are chances that maybe it will not code for that amino acid or it will code for some other amino acid. This information is very important. Now, you had this sequence in DNA. It was a normal sequence. So this was a normal gene. Okay. Now, what happened? By some error, this G, it got replaced with A. So earlier, what was present there? It was a G which was present over there. Guanine was present there. It got replaced by adenine. Now, the same gene has undergone mutation. Which type of mutation? Substitution type of mutation, because guanine is being substituted by adenine. Okay, or we can say for example, this is thiamine. So earlier what was over here? Thiamine was over here. This got substituted by cytosine. So a kind of change. if something like this is happening to the sequence of your nitrogenous bases in DNA, this type of thing, this type of change, this type of error, this type of alteration is called substitution. Okay, my dear students, now there is one important term. Substitution could be of two types. It could be transition substitution number one, or it could be number two, either transition substitution or you will have transversion substitution. Really simple. So substitution itself is easy. You have replacement of one nitrogenous base with some other nitrogenous base. Okay. Now it has two types: transition substitution, transversion substitution. Pay attention. You know, according to CHROMOSOM's rule, adenine always pairs with guanine. Guanine or something else? I'm wrong. So adenine is going to pair with what? A pairs with T, G pairs with C. This is the basic thumb rule which you people know from class 11. Okay? A pairs with T, G pairs with C. A and G are what? They are purines. T and C are what? They are pyrimidines. So you have how many types of nitrogenous bases in DNA? 4. Adenine, guanine, thymine, cytosine. 2 are purines, 2 are pyrimidines. They have 2 rings, and this just has 1 ring. Okay. So this is the basic thing. Leave it. Now for the time being, we need this table to understand transition and transversion substitution. What happens in transition substitution is that a purine is replaced by a purine, or a pyrimidine is replaced by a pyrimidine. This is happening in transition substitution. Means what? Simple. Suppose this is thiamine. What is it? A pyrimidine. It's a pyrimidine. It will be replaced by cytosine, not by A and G. So this will be replaced by what? Suppose this is being replaced by cytosine. Let me show you over here. I'll take a different sequence. So I have a sequence like this. It's very simple. CTG, CCT. I have a sequence. In this, I will take, suppose, T. I will replace T with some other sequence maybe. I will write down in place of T. In place of T, I will write down C. The rest is same. This type of mutation is called transition. Or I will write down in place of guanine. Guanine is what? Purine. I will replace this guanine with adenine. So a purine is replaced by a purine. A pyrimidine is replaced by a pyrimidine. This type of mutation is called transition substitution. What happens in transversion? A purine is replaced by a pyrimidine. Simple. Or vice versa. A pyrimidine is replaced by a purine. Say for example, I have T, C, G, C, T, G and C. Okay. Now you have T. What is it? Pyrimidine. It will be replaced by a purine. Suppose in place of T you will have A now. Okay. Suppose you have C, or we'll keep C as such. You will take now G. G will be replaced by T. Observe this. A pyrimidine replaced by a purine. A purine replaced by a pyrimidine. This is also substitution because one base is getting substituted by other. But this type of substitution is called transversion substitution. You will see ahead when we will discuss this disorder called sickle cell anemia. In this disorder, you will see that a purine is replaced by a pyrimidine. In this case, you will see adenine being replaced by thiamine. So in normal conditions, we have a sequence like GAG. Okay. But in case of sickle cell anemia, the sequence changes to GTG. Okay. So because of just one base getting substituted, adenine being replaced by thymine in DNA, a disorder arises, a genetic disorder arises in which your hemoglobin becomes defective, and the condition is called sickle cell anemia. We will discuss this ahead. But what is the reason behind the sickle cell anemia is substitution. And which type of substitution, to be very specific? It is transversion substitution, where adenine is being replaced by thymine. Okay, this was all about your substitution. Now let us go to deletion maybe. Okay. Deletion and insertion will be somewhat similar. See, in case of deletion, number second is your deletion. And what will happen to the gene? A particular nitrogenous base will get deleted. Okay, it's very simple. Let me show like this. We have a sequence C, T, G, C, T, C, 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 A. Okay, this is a sequence in a gene. Now, in case of deletion, you will have what? You will have removal of either one nitrogenous base or a few nitrogenous base from a particular gene. Say, for example, I'll do like something like this. I'll say, suppose this T got removed. So what are we left with? C, G, C, T, C, 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 A. So if you remove T, you're left with C, G, C, C, G, C, T, C, 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 A. Okay. So if you'll observe, the information will not be read in the same manner as it was read earlier. Earlier, the codon system was like this. Earlier, it was this codon followed by this codon followed by this codon. Now the sequence has altogether got changed. Now you have a different pattern of codons. You have now CGC, TCC, CA. Similarly, the change will be till the end point of this gene. So entire information will be changed in case of this deletion. Similar is the case with your third type, that is insertion. See, it's a similar type of change. I'll take the same gene into consideration. Now in this case, earlier I told you that this will get deleted. The consequence will be this. Now I'll say, my dear students, let us say in case of insertion, a base gets added over here. Okay, so what will be the definition of insertion is addition of one or few nitrogenous bases. Simple. So I will add, suppose one base, suppose I will add over here guanine. What will be the condition? Now you will have C followed by T followed by G as usual. Then you will have this new G which was inserted right now, the new G. Then that will be followed by C, T, C, C. Then you have C, over here G, then it was C, T, C, then C, C, A. See now what has happened to the sequence? CTG is as such. This is the new base which was added, G, which I added over here in case of insertion. Now the new sequence, it was to be CTC, now it is GCT. Third was supposed to be CCA, now it is CCC. Same way other sequence will also be disturbed. That means the entire gene sequence gets disturbed. In case of what? In case of both deletion and insertion. Okay? So the entire frame of the gene, which is actually read by your RNA polymerase during the process of transcription, that entire reading frame is getting changed. Such changes are called FRAMESHIFT mutations. They are called FRAMESHIFT mutations. Pay attention what do I mean by over here? Can't you observe that the reading frame of the gene is getting disturbed? This, which I will encircle, was the original reading frame of the sequence, where we had 3 triplets, CTG, CTC, CCA. This was the normal reading frame of the gene. Now, if you talk about deletion, if you will delete T from it, you will get a reading frame like this, which is totally disturbed. If you will insert some sequence, you will get a reading frame which is like this. It's also disturbed. Now, whatever is the polypeptide which was coded by this original gene, either that polypeptide will not be formed, or either if it be formed, it will not be having the correct amino acids. Chances are different, but we have to focus upon two things. Either now the gene will not be expressed, either the gene will become faulty, either the product will not be obtained, or if transcription will happen, RNA polymerase will come, these codons will be read, the final end product will be a non-functional protein, to be specific a polypeptide. Then that is going to result in different kinds of genetic disorders. Such a type of mutation which happens due to deletion and insertion, such mutations are called frameshift mutations. You have another term for these mutations, you call them gibberish mutations. I'll make you understand this by taking an example which is very simple. Okay, I will make you understand frameshift mutation or gibberish mutation by taking a simple example, in which I will not be showing you this codon system, original codon system. I'll be taking some phrase into consideration, some English sentence I'll take into consideration. Suppose, my dear students, will say that why did cat eat rat? Let us take this into consideration. Isn't it a statement? And in this statement, when we say why did cat eat rat, every 3 letter is making some sequence. It's making some sense. See? Why did cat eat rat? Same is the condition with your DNA. In DNA, 3 nitrogenous bases at one time, say for example C, cytosine, guanine, thymine, they make up one sense, they make one codon. Okay, so 3 bases make one codon, and one codon codes for an amino acid. Now, if something will happen to the codon, correct amino acid will not be coded. At certain times, some other codon is generated which may code for the same amino acid. That is secondary thing. But if something will happen to the codon, maybe this amino acid will be replaced by some other amino acid. That's what we call mutation. Let me make you understand over here. Say for example, in this example, let us do one thing. Let us remove D. Okay? If we will remove D, what will happen? How will we read then this? Y will remain as such. Remove this D. You will be left with I, D, C. Then A, T, E. Then A, T, R. Then A, T. Is it making any sense? You have this making sense. Does it make any sense? 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 No. What has happened? We have done what? We have done, in this case, we have done deletion of just one letter. We deleted one letter. Same is happening to DNA. If you are deleting some nitrogenous base, sequence is getting changed. Frame is getting changed. We call this change what? We call this mutation frameshift mutation. Let us insert something. Over here, let us insert, say for example, X. Let us insert X over here. Okay? What will be the condition now? You will have W, H, Y. Okay? So for example, first combination doesn't get affected. Next, X, D, I. Next, D, C, A. Next, T, E, A. Next, T, R, A. Next, T. What is happening? Again, the frame is getting changed. This happened when we did what? Insertion of X. So both in case of deletion or in case of insertion, your entire frame of a gene is getting changed. This reading frame is getting changed. That's why we call it a frame shift, or we call it a gibberish mutation. So such mutations can happen in your DNA. The same type of mutations is happening in Duchenne's muscular dystrophy. It's a disease which we have to understand in case of genetic disorders. In this disorder, the same kind of mutation is happening to the dystrophin gene. Similarly, you have cystic fibrosis, you have Leigh-Schneihan syndrome, in which same type of frameshift mutation is happening either by deletion or by insertion of just one nitrogenous base. Okay, in this way your genes are getting mutated. So I gave you the concept of mutation by substitution, either by transition substitution or by transversion substitution. Then I gave this concept of mutation by deletion, mutation by insertion. And then you came to understand about frameshift or gibberish mutations, which are very lethal and which are very harmful to the body. In Dutch muscular dystrophy, you will see that dystrophin gene will not be produced and the person will die coming to the approaching age of 20 years. The person will die because of these defects, because of this minor change in the gene sequence. Now there are three terms which you have to understand, because I told you sometimes you will get a different codon, but the same codon will code for the same amino acid. That's called same sense mutation. I'll give you at the end of this lecture, I'll give you now the definition for same sense, missense and nonsense mutation. Let us take up that. So that is very important, and you will not be able to understand those three terms separately. You will be able to understand that within the same topic. They are very simple. I'll make you understand. So three terms are there, my dear students. In order to make you understand these three terms, I will take into consideration the same sequence which I told you earlier, which was GAG. If you remember, I told you earlier that in case of sickle cell anemia, GAG is replaced by GTG. Okay? See what is happening? If this is replaced by T and it remains the same, GG. In this case, earlier this codon was coding for this amino acid, glutamic acid. Now after change, after this substitution, specifically transversion substitution, it will now code for valine. So what is happening in this case? Simple. Codon is also changing, and amino acid which was to be coded, that is also changed with a new amino acid. Now let us take a condition. I'll take the same codon GAG. What is it coding for? It's coding for glutamic acid. Now in this codon, we will replace one base. I'll be showing now G, A, and in place of G you have again an A. So you got this replacement done. Earlier it was GAG, now it's GAA, but the condition over here is that just this codon GAA also codes for glutamic acid, the same amino acid. So you have observed in this case that codon was changed. So codon was changed definitely, but amino acid is same. So the same amino acid will be added to the polypeptide chain. This is the fun of degeneracy. Genetic code is degenerate. For one amino acid, you could have more than one codon. So this is the benefit. So see over here, mutation happened, but it doesn't lead any effect on the amino acid it coded for. This is one of the advantages of genetic code being degenerate. Similarly, I'll take one more case, the same codon into consideration, GAG, which was supposed to code for glutamic acid, an amino acid. Now a mutation is there. You must know that the strand of DNA which I am showing over here, the codons, they are present in which strand? They are present in coding strand of DNA. Okay, you will find the same sequence in which strand of DNA? You will find the same sequence in coding strand, as well as the same sequence will be in the mRNA. Okay, so over here I will show mutation as per mRNA. In mRNA, say for example, you will see that over here it's not a thiamine, it should be actually guanine in mRNA, but a mutation happened where guanine was replaced by thiamine. In mRNA we don't have thiamine, so I am showing that with the help of uracil. So UAG was the product in mRNA. Okay, UAG is what? UAG is not coding for anything. It's a stop codon. Okay, so some functional codon, some functional codon was replaced by a stop codon. Stop codon is also called which codon? It's called nonsense codon. Okay, so over here what has happened? It's a drastic change. You have codon getting converted into a nonsense codon. Think about this case. Say for example, this was the original gene and over here there was your stop codon. And over here there was this normal codon. But this normal codon got converted into what? Stop codon. The position of stop codon is actually over here. But now stop codon arose here because of mutation. After transcription you will have an mRNA only of this much length. Okay, so this mRNA will give the rise to a short polypeptide which will be non-functional. Okay, now look at these cases. This is the first case. Okay, this is the first case where one codon got replaced by another codon, which resulted in total replacement of one amino acid with another amino acid. Such type of mutation is called a missense mutation. This is called missense mutation, where one codon is replaced by another codon, one codon is replaced by another codon. Simultaneously, one amino acid is replaced by another amino acid. Okay, this is the case you will see in sickle cell anemia also. In this case, you will see one codon replaced by another codon, but the amino acid remains the same. This is called silent mutation because it will not pose any threat to the organism. It's silent. Okay, because information is going to be still same after mutation. You call it silent mutation or you call it same sense mutation. Pay attention, very important as far as competitive examinations are concerned. You are often asked about this tricky question out of which you have been given statements and you have to select, is it a missense mutation, a same sense mutation? What do you call this thing? Where some codon is getting converted into a stop codon, into a nonsense codon, you will call it, accordingly you will call it a nonsense mutation because it is changing the sense of your codons. It's making your codons nonsense. So you have three more types of mutations. Number one, missense mutation. Number two, same sense mutation. Number three, nonsense mutation. Very easy. One codon replaced by another codon. At the same time, one amino acid replaced by another amino acid. One codon changed by another codon, but the amino acid remains same. Degeneracy concept. One codon replaced by a stop codon, making a nonsense mutation. Okay, my dear students, all these examples fall for your point mutations. They are all your point mutations because they are happening at one place. Okay, they are happening at one location, one specific point. Similarly, you could have 10 or 20 or hundreds of mutations happening in just one time. That mutation is not so common. Those are rare mutations, but they also happen. You will also find those mutations taking place. Those mutations are called, my dear students, they are called gross mutations. Okay, try and understand. Point mutation, where you have change in one or two nitrogenous bases, like these cases. We have gross mutation, where you have multiple mutations happening at same time, maybe couple of 10 or hundreds of bases changing at a same time. That case is rare, but that also happens. This was all about gene mutations. Inshallah, in next lecture we will try and complete our chromosomal mutations. We will discuss genomic mutations as well as chromosomal aberrations and their subtypes in next lecture. Till then, watch this video, take up all the information, try and clarify your concepts. If you have any questions, drop me that question in the comment section below. Don't forget to like, share, comment and subscribe to my channel so that I can reach to more and more students. Thank you so much for being with me. Take care. Fi amanillah.